Hey guys. All right, we are doing our DNA review. So let me get this started. All right. Our DNA is our blueprint, and that is our first slide here. So this is the instructions of who we are and what we are, everything about us. DNA stands for deoxyribonucleic acid. You should be familiar with this. It is found in all cells packed tightly within the chromosomes in every nucleus of every, and this should say cell. Every cell that we that we have contains DNA, and all of our DNA is exactly the same individually for us. Segments of DNA code for traits, what we call genes. So a trait might be something like hair color. That's a trait. And then we have something called alleles, which we're going to get into, and we have two alleles for every trait. But so traits are so hair color, skin color, height, all of those are traits. So chromosomes, if you remember, we have 46 chromosomes. We got 23 from our mom and 23 from our dad. So DNA is the molecule of life. It's wound into this little shape right here, and you should be familiar with that already, and that's called a double helix. The structure of DNA was established by Watson and Crick. Were credited with it. Rosalind Franklin, which she is pictured next. She took a picture of DNA using X-ray diffraction, and they actually used Watson and Crick used her information to help them develop the shape of it, and they were credited for it, and they won the Nobel Peace Prize. She did not get the Peace Prize because she had already passed away. There's actually a story on that, and you can Google the um, history of DNA, and it's on YouTube, but it's a pretty interesting scenario and situation. So Rosalind Franklin took a picture using X-ray diffraction. DNA is made of repeating subunits called nucleotides. A nucleotide, if you remember, is a sugar phosphate and a base. So a sugar, a phosphate, and a base. We have a song. It's "We love DNA made of nucleotides, a sugar, phosphate, and a base bonded down one side." So a nucleotide, I want you guys to remember sugar, phosphate, and a base. The nucleotides are four bases: adenine, thymine, guanine, and cytosine. The bases are also called nitrogenous bases. This sounds like a big fancy word, but it just means it's made up of like a lot of nitrogen. And we need nitrogen in our body because it's in our DNA. So the nitrogenous bases are ATGC, and they always bond together like that. Because if you can see this picture in this image, there's like they're partnered together. It's a double twisted ladder, which is called a double helix. So on one side, you have like guanine. So if this is guanine, then this side over here would be cytosine because GC always pairs together. I like to remember this. I would say at Greene County, and that's how DNA always pairs together. So if I were to say like, let's do like a little cheer. I would say A, you would say T. I say G, you say C. I say C, you say G. So DNA is made of nucleotides. What's a nucleotide? A sugar, phosphate, and a base. The bases are these. The phosphate backbone is a PO4 group, a PO4, and that's this. And the sugar is over here as well. And the sugar in DNA is, any guesses? It is deoxyribose, and it ends in ose. And if you remember, sugars typically end in oses, and it's deoxyribose. So a closer look, three parts: phosphate, deoxyribose sugar, and the bases. The phosphate, the sugar, and it is a pentose sugar. So it is a pentagon shape. So pent means five, phosphate, sugar, base. DNA has millions of nucleotides. So there's millions of these that are in our body, and actually in every one of our cells. So there's millions of nucleotides. Let me move myself over. So you definitely need to know this structure because you may see something similar on the EOC and you have to label like what is the phosphate, what's the sugar. And if this one right here is G, you have to know that this one is C. Something else I want to point out here. Notice there are three bonds in between the G and C, and only two bonds in between the A and T. And these are actually called, and I believe I have them somewhere on the slideshow, we have purines and pyrimidines. And I like for you guys to remember purines are the first two, so the A and the G, so at Greene County, the first two purines, so adenine and guanine. So There's two of those, and there's three pyrimidines. That's a longer word. And there's three, and that's uracil, because that's an RNA we're going to get to, thymine, and cytosine. So adenine and guanine are purines. Thymine, cytosine, and uracil are pyrimidines, and that's you're going to see that later, but you could go ahead and write it down now. But notice here, these have three bonds in between them, and these have two, because that will be critical when you label that on the test or on the EOC. Have to identify what is what. One thing also to just remember here is G pairs with C, T pairs with A always. DNA contains the instructions to build an organism. Could we clone organisms if we have their DNA? Absolutely. So, is it okay to clone humans? I don't know that we're necessarily there yet. We'll debate that in class. Are we using GMOs, genetically modified organisms today? Yes. Are we cloning animals? Yes. We already are. We're doing that. The first organism to be cloned was Dolly, and that was the sheep in 1996. And you may already know that. Hopefully, you've learned that already from a science class previously. This is the base pair rule, ATGC. And notice here, okay, the three bonds here and the two bonds here. Three bonds here and the two bonds. The sides of the DNA ladder are the phosphate and the sugar. Chargaff's rule. This is ATGC. Now we can make this, and Chargaff was another scientist that he found out that proportions of A is equal to T and G to C. So you could actually have like a math-like question on this. And I'm going to give you just something to think about. So if we had 20% adenine, how much thymine would we have? Well, if A is 20%, then this should be 20%. And if A plus T, that equals now 40%. So how much of these two should be left? And that should be, if this is 40, then this should be 60. So 60 divided by 2 would be 30 and 30. And we will go over that in class as needed, but that's something to know. So let's do another problem here. Let's say we have 10% cytosine. How much thymine would you have? I'll give you a second. This is 10%. Well, if this is 10%, then this must be 10%. 10%. So that's 20%. 100, because it's out of a total, 100 minus 20 is 80. So this is 40 and 40. Applying the base pair rule, let's go ahead and finish this, and you should write this in your notes. Make sure you can do this. If one side is this would be also called the template strand of DNA. So if this is A, this is going to be T, A, T, A, G, T, A, C, G, C, C, C. Make sure you understand that. The central dogma of biology, that means the basis of biology. You have to know that term. I don't know why they like to use it, but they do. But it is DNA, RNA, protein. So if you ever have to fill in that blank and one of these is missing, it's DNA, RNA, protein. What goes along with this, and I've put these terms here, replicate is DNA. That is always associated with DNA, and that is using ATGC. RNA, from the DNA, when we create the mRNA, that is transcribing or transcription, and that's AUGC, because RNA only has U. We're not there yet in the slideshow, but I just want you to know where we're going. And then lastly, protein. Proteins are made of amino acids, and that's dealing with translation. So it's DNA, RNA, protein, replicate, transcribe, translate. You got to get that into your brain. Make it a cheer. Replicate, transcribe, translate. Replicates when DNA copies itself. Transcription, RNA is created from DNA. Specifically, this is the mRNA, which is the messenger RNA. It carries the message. And translation, this is when codons are created and then used to form amino acids, which in turn create proteins. Amino acids, remember, there's 20 amino acids. These make up proteins. What are proteins? They're enzymes. They're catalysts. They're in every bodily function. We must have those in our body to control all of our metabolic processes. DNA replicates. Remember, this takes place during interphase, specifically during the S phase. And remember, you should be familiar with all of this stuff already. This is going to be our interphase, which is G1S, G2. This is prophase, metaphase. If you remember, these chromatids are pulling apart right here. That's anaphase. They are now moving to the poles, and that's going to be telophase. And then lastly, split into two different cells, and that's cytokinesis. All right, we went over these in class. Make sure you can answer these. I'm not going to answer these for you. Make sure you can do it. DNA is semi-conservative. You have to know these terms. That means for every time that it replicates, you have every strand has an old and a new strand. DNA is complementary. It means each nitrogenous base is partnered with its complement. Like if I say A, what's its complement? It's T, G, and C. Anti-parallel means it runs opposite. If this is the 5' end over here, then this would be the 5 or the 3' end. And it runs 5' to 3' and then 3' to 5'. So if we ever have, and we'll show you this in class, 5' here on this side is going to be 3'. And you need to be able to point those out. It runs opposite. Anti-parallel. So DNA is semi-conservative, and this shows you the old and the new. Old strand is here. It opens up, and then the new bases come in, and this means it's semi-conservative. You are saving, kind of, okay? The new bases come in and fill in, and then you have an old strand and a new strand. Getting the message out, DNA stays in the nucleus. It is used to make RNA. We just went over that. RNA contains the code to build proteins. RNA leaves the nucleus and heads to the ribosomes where proteins are made. Remember, ribosomes do protein synthesis, and this is the central dogma. Super important. Now, let's do a quick review. When DNA makes a copy of itself, replication. Then when the RNA is formed, what do you call that? Replicate, then transcribe. And then lastly, to form this is translate. The central dogma, here we go. It shows you what's going on here. Replicate, transcribe is happening here, and then translating. RNA is the messenger. Single strand contains ribose, sugar, contains no thymine. It has uracil instead. Follows the base pair rule AUGC. And this is the messenger RNA only that we're talking about right now. Well, I take that back. All RNA is this, but when we form this strand, this is the mRNA when it's formed from the DNA. Single strand contains ribose, no thymine. So when we do this, this process now would be called transcription. So it would be AUTAAUGCCGCTAGCAU. So I'm going to do that one more time. So look at the DNA. Remember, RNA doesn't have a T to use. So UAUCGCACU. That is transcription. Then from here, I'm going to put myself way over here. Okay. The RNA carries the message to the ribosomes where the proteins are made. And here's the 3 types of RNA. mRNA carries the message, tRNA transfers the message, and rRNA helps make the proteins. You need to know what these letters mean. M is messenger, T is transfer, R is ribosomal. And here's the types of RNA and what they do. RNA is single-stranded. Quick review over what is a gene. Okay, remember a segment of this are the traits. How does DNA make a protein? I apologize. The letters are the code, which tells the cells to know which proteins to build. Every 3 letters are called codons. For example, AAA specifies a single amino acid. So you take 3 letters and you put them together, and that makes a amino acid. And we're going to show you how to do that, but you have to understand how these codons are made. So first it's DNA, RNA, protein, replicate, transcribe, and then to translate, you take 3 letters, you put them together, and that forms a codon. And then you have to be able to read a codon chart. You don't memorize it, you utilize it. Why are amino acids important? Remember, these are protein. These build proteins. They're the building blocks that make proteins. Some are functional, like lactase breaks down lactose. Remember, if it ends in ASE, typically it's an enzyme. So lactase breaks down lactose. If your lactase doesn't function properly, then maybe you're lactose intolerant. Some are structural proteins that make up muscles and blood cells. So proteins control every metabolic function in our body. Proteins are made of chains of amino acids. There are 20 amino acids. How to get from a gene on DNA to a protein. Step one, the DNA must be made into the mRNA, and that's through transcription. DNA strand is this. From that, we make the RNA strand. So your challenge is to do this in your notes. I'll give you just a second. Put this in your notes. What pairs here to form the mRNA? All right, here you go. U, 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 G, 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 U, A. Hopefully you got that correct. Next, the ribosomes RNA travels to the ribosomes. Ribosomes use the code on the mRNA to build a chain of amino acids. This is called translation. Transcription and translation here. So first you replicate, then you transcribe to make the mRNA, then you translate. So you use the 3 letters to form codons, and then you make a protein, the protein. Here is the codon chart. I'll show you how to do this in class. And Miss Nolly will show you how to do in this class. The way that you read this, and there's 2 things that you need to look at, the wheel and then knowing this chart, because there's 2 things that the EOC could give you. The way that you read this is the first letter. This is called the first base over here. We always start here. And then we read the second letter or the second base, and then the third letter. So what amino acid is made from AAA? So we start here, and it's A. This means it's only going to be in this section here. Okay, AAA. The second letter is here, A. So A, and where do these intersect? This A is here, and this A is here. So it's going to be right here. It's going to be one of these. And the last letter is an A. So we go to the third letter, and so it's in this, and then this one actually here, and this is called lysine. Now for you, because all this says is LYS, you know, lyse. And this is lysine, and that is the amino acid lysine. There you go. So now you try U. Okay, U. So we know it's going to be in this section. Second letter U. And the third letter is U. It is, and this is actually the big one, phenylalanine, but all you know is PHE. All right, let's try another one. I want you to try CAG. CAG. All right, C is here. A puts it here. Are you following me? And then G puts it here. So, GLN. That's what it is right here. And this would be our answer. GL. It actually, so yeah, CAG. There we go. All right. What if a mistake is made when DNA is replicating? Well, that can happen. And then we get something called mutations. There is a deletion. If you delete something, what happens? A part goes away. Well, that actually causes a frame shift. That's really, really big. It's not just a point mutation. A point would be only on one little section. So imagine this one, and I'm sorry, do deletion down here. If you delete one nitrogenous base, it's going to cause everything to shift down, and then everything behind it is going to be different. Is that a problem? Yes, that's a big problem because it's going to cause everything to shift down. If you have just a point mutation, something like a duplication. So we just change one letter, and let me see, this is a substitution. Here's one. It's a point mutation. The letter's different. Just one letter changes. Is that a problem? Probably, but it doesn't have to be. It could be silent. But it doesn't cause a frame shift. It only causes one issue. So that's what we look at here. So we have deletion, which is deleting one. You could have an insertion. That's actually not on here. That would be adding one, which should cause a frame shift. Duplication, that means a chromosome or even a nitrogenous base is copied. Translocation means move one to another place. Insertion is pictured down here. Translocation means if I were to take one from over here and I were to move it over here, would it affect all my codons? It would. Now, a silent mutation means sometimes I might change a letter, but it codes for the same amino acid. Now, this is confusing. We're going to go over this in class, but I want to go back on this. Let me see if I can go back. So, say, for example, I only changed one letter. There's more than one way to get arginine right here. So, it could be a mutation, but it actually led to the same amino acid. Therefore, it was silent. So, a missense and nonsense are also mutations, and we will look at these in class. All right, hemoglobin, this is a real life example, is a protein that makes up red blood cells. The DNA, your DNA tells your body what kind of cells to make. Well, our red blood cells should be like circular shape. That way they can carry the maximum amount of oxygen and hemoglobin. But sometimes they become crescent formed. And that is a mutation. And that leads to something called sickle cell anemia. And we'll get to that one in just a second. So, how does a mutation a mutation is a change in DNA. So, what does it do? It just changes one. A point mutation is just changing one to another. Remember, that's just one place. So, single base. Sometimes the whole chunks of DNA get altered. Either way, it's going to function the gene and the trait. I want to point out all mutations are not bad. They could be good mutations. They could be not recognized because they could also just be silent. So all mutations are not bad. All right, back to sickle cell. This could clog up throughout our entire body. We have valves. Those sickle cell and those crescent shapes get stuck together. And what they can do is they can clog an artery or a vessel, and then it could cause pain. So someone with sickle cell, and it does run in different nationalities, but it is a problem. It is something you would live with for the rest of your life. It is symptoms are anemia, which is someone who just has like low iron, pain, swelling of hands and feet, delayed growth, problems with vision. What causes it? We have this, and you can read Alexandra's story, and then there is a video on this. I just deleted it. I wasn't sure if you guys would be able to see it, but this is just a real life story of her sickle cell disease. A change in DNA changes the protein. So these are mutations, and this is actually what's happened with the sickle cell. So you have the DNA, this is 3' and this is 5'. This formed the mRNA. See, this is normal, and then this is the sickle cell. Notice the change right here. And this is the U, and instead it actually should have been GAG, but instead it was GUG, and that is the problem right there. And instead of coding for GLU, which I think is a glutamate, it coded for valine, which is a problem. And now instead of creating this type of blood cell, it creates this one. What happens when your DNA changes? Can it be passed to your offspring? It can. So this can cause a disease that can be passed to offspring. Genetic diseases are not contagious, but they can be inherited. So this shows you, this is called a pedigree. Half shaded means a carrier. That means they have it, but they don't show it. So these two parents have it. They pass it to one child. Two children are carriers, and then you have the non-carrier. Some examples: sickle cell, cystic fibrosis, Huntington's, dwarfism, and muscular dystrophy. Sickle cells get stuck, and this just shows you an example. Sickle cell disease, some more information. Why don't Alexandra's parents have the disease? And your answer here would be because they're carriers. So in order to have it, both the offspring needs to not just be a carrier, they need to have both copies of the mutated gene. And that is our last part. Thank you.